All right, so now let's kind of sum up what we've talked about so far in the world of DNA. So we've talked about one strand basically having these bases that stick up, and this one strand is written from the 5' to 3' end, and then we get a complementary strand that comes along with that. It runs anti-parallel, which is what we learned in the last video. So that means this is the 3' end, and this would be the 5' end, and then we have bases that complement the ones that are there already. So A to T's and G to C's. We also have now talked that this is kind of the ladder. That is what we refer to as the DNA ladder, because if I connect these two pieces together, you can kind of see how that turns into a ladder. So over here to the side, I could represent a DNA molecule kind of like that. All right, so we've talked about some of the dimensions now of the DNA ladder, and one of the dimensions is that it turns. So if I look down the actual tunnel of the DNA, we are going to see it corkscrewing around and around and around. So we actually will see this helix begin to get formed, this slinky. We've talked about the helix before in the amino acid and protein lecture. So the same thing happens here with DNA, is that this DNA molecule looks down, if you look down a tunnel, and it will turn, and it turns into a corkscrew. And this turn is a right-handed turn, and the turn ends up giving me this thing that we call a helix. So helix is going to come back when it concerns the DNA field. Okay, so after this, after we talk about these strands and complementary strands and the ladder turning and corkscrewing, we also talked about this idea of a major and minor groove. So major and minor groove. And the major and minor groove, you can almost think of it as something like here. I mean, you see those little notches that this helix will make? Well, those notches are very important. So I'm just going to label these as notches. And these notches or these grooves on the surface of the DNA are actually kind of important because proteins and other molecules can bind to these major and minor grooves. So this is a way of associating it with the DNA molecule, maybe having some interaction between a DNA molecule and something else. But this is an activity site that we can talk about. And the major groove, of course, is a larger notch than the minor groove. So we need to take a look at maybe a better picture of the major and minor grooves. And in order to do that, we need to take a look at a space filling model. And that space filling model can be found in your PowerPoint. So let's take a look at the PowerPoint slide, and let's take a look at the 3D structure of this DNA and talk about the major and minor grooves. All right, so here we go. This is what we're talking about. And this space filling model is going to kind of condense things down for you a little bit. And if you take a look at the key, you're going to see it's capped out on your screen. But the small little white ones, of course, are hydrogens. And then the red ones are oxygens. And the blue ones are nitrogens. And then the black ones are carbon. And, of course, the yellow ones are our phosphorus. So we've talked about this idea that bases are on the inside of the corkscrew. So when these two strands turn on each other and make this helix, the bases on the inside are flat, and they make these runs all the way up and down the DNA helix. Okay, so that's where our bases are located. This strand on the outside, this ribbon kind of, this ribbon is where your phosphorus is going to be located. That's where your sugars are going to be located. And that's also why you see a lot of the reds or the oxygens up and down this backbone, because those oxygens are making up the sugar molecules sometimes. All right, so we looked at this diagram because we wanted an idea of what a major and a minor groove is. And here it clearly shows what the major and minor groove is. When this DNA strand begins to turn on itself like this, you're going to see a major groove. So if you take a look at from when it turns, if you take a look at this cavity or this empty space that's here, this notch that begins to happen, this is a very large notch. So if this was kind of a cliff and this was a cliff and this was a valley here, then you would have to jump quite a bit of distance to go from one cliff to the other, right? And that's something that I probably wouldn't want to accomplish at all, or I might hurt myself. So that's what we call the major groove. Now the minor groove is very similar. It goes from one peak to the next, and then there's a valley in the center. But this notch or cutout is not as pronounced as some of the others. So because this isn't as deep or maybe as wide, you're going to see that they call this the minor groove. So we get a major groove and we get a minor groove. Slightly offset a little bit, but the total width of the DNA strand from top to bottom is basically the same all the way through. So that is what we talk about when we talk about major and minor grooves. A lot of people get confused with it. They don't really see it. If you look over on this side, this is going to be the major groove here, and then this one's going to be the minor groove. So again, what we see is this whole complementary aspect to the DNA structure. We see a major groove on this side, and then on this side is a minor groove. And then the same thing happens up here. A minor groove happens here, so on the other side, a major groove is going to be located there. So that's how you determine the major and minor grooves. In the PowerPoint, you are also going to see a slide that looks something like this. And if you activate it, it will actually begin to turn the DNA molecule for you. So what you will see here is this DNA strand, and they're just turning it. So you can kind of see how this thing turns like a slinky. You can also see that in the center here, these are your bases, and they stay flat all the way up and down the DNA molecule because they should. That's our stepladder, right? And then the same thing happens here. You can see the oxygens on the sugars and the phosphates that make the backbone up. So just a 3D representation of the DNA molecule itself. And this is what Watson and Crick basically proposed and discovered during that time. And that is why we call it one of the most sounding alarms in biology and biochemistry and chemistry in general that was discovered back into the 1950s. So not too long ago. So again, we've came a pretty long way since that point of discovery. But this shows you the work that Watson and Crick really put into this whole description. Now, how did they determine that this thing was a coil? Or how did they prove that the thing was a coil, right? Just because you think something doesn't mean that it's true. You have to have proof. So you have to have concrete proof of what they think is going on with this DNA molecule is really the actual structure of it. And that proof came from, again, X-ray crystallography. So we talked about this whole thing about X-ray crystallography. There's another representation of a major and minor groove. But this X-ray crystallography basically allowed us to look at a shape and determined that this was, in fact, a helix. So what happened is that a sample of DNA was analyzed on this X-ray crystallography machine, or this instrument, and this is the picture that these people would see. Very, very similar to maybe an X-ray that you would take in a doctor's office. And Watson and Crick, neither one were really experts at the time on how to interpret this information. And Rosalind Franklin, who was a woman, was actually able to take a look at this and say, guys, I think you're onto something. I think that this X-ray crystallography shows that you've got a coil. Something is circular in nature, because we take a look and you can kind of see this axis here, and you kind of see the circles that happen one after another, after another, after another. And it's almost like looking down a tunnel. So because of that, they get this proof that DNA actually does coil on itself, and it actually does form this tunnel that we can talk about. And this helped prove the 3D structure of DNA, but it was Rosalind Franklin that did that. So what began to happen is that Rosalind Franklin began to interpret this information. She began to write down the information in the lab book. And then one day, Watson and Crick come into the laboratory, and they basically want some of the interpretations that Rosalind Franklin did. And Rosalind Franklin's lab book was left in the lab, and they took it. And they took the lab book, and they took her interpretations in the data, and sure enough, it proved out to be true, and they used this to actually prove the 3D structure of DNA. The sad part about it is that Rosalind Franklin was not there the day that they took that lab book. Not only that, but Rosalind Franklin also passed away due to the X-ray that she was exposed to during her time in the field of X-ray crystallography. So she did not wear protection. This was really before the dangers of X-rays were really kind of promoted or encouraged, as far as self-protection equipment, personal safety protection equipment. And Rosalind Franklin did pass away before even she knew what was happening or the importance of what was going on during that time. So Franklin never protected herself from the X-ray sources. I don't think that many of you will work with X-ray crystallography machines, but if you do, keep that in mind. You're exposing yourself to this source on an everyday basis, and that's something that you do want to take care of. And she, again, died without knowing the role that she played in determining the structure of DNA. And over a course of time, her name was forgotten, her name was dropped, her name was not mentioned, and it's just really Watson and Crick that is given kind of the rights to this whole discovery. There's actually a fourth one too as well. Wilkins is going to be another one that's thrown into the bunch. He actually helped out with this whole study as well, and that's another name that gets dropped most often from when we talk about the 3D structure of DNA. It seems like we always talk about Watson and Crick, and that's it. So here's Rosalind Franklin, born in 1920, died in 1958. So she was 37 when she died. She was a biophysicist and chemist. She was specially trained in X-ray crystallography. Again, it's very nice to see a female face for a change that pops up on the screen, because most of the time these have been white, older men that we have seen. And now finally we get a woman that we can talk about for a change. So she recorded the picture of DNA with another researcher. The researcher showed the picture to Watson and Crick. Franklin did not know about it. She did not give them permission. She wasn't there when it happened. The other researcher, named Wilkins, Watson and Crick, was awarded the Nobel Prize, and Franklin was not. Yeah, that kind of sucks, doesn't it? Here you've gave basically these people the pathway to get a Nobel Prize, and your name is hardly ever mentioned throughout history as an important player in what she did. And she ended up dying of ovarian cancer. And now, since we've talked about our first woman, let's briefly mention our first African American chemist, and this is George Washington Carver, 1864. So finally, we see a minority or an underrepresented person in the science field. Doesn't have anything to do with DNA, but I think it's just kind of nice to see an African American showing up as well in the actual chemistry field. And George Washington Carver was all about peanuts. So you can read about him, see what he did, but that's Mr. George Washington Carver.